channel Pathology Vision. Myself a plant research scholar. So in the previous video I have solved the question paper of I.A.R.I 2011 from question number 30 to question number 100, and the remaining questions will be solved today. So watch the video from beginning to the last and subscribe to my channel if you like the video and have some helpful knowledge. So now we will start the question solving from question number 101. So the question number 101 is more number of spots of Manichuria in Achilles causing scab disease is discharged during the day than in night because. The question is about the Manichuria in Achilles. So the answer is leaf wetting is an essential for the spore release. In Manichuria in Achilles, that is causal organism of apple scab, the major criteria of this one is the leaf wetting is essential for the spore release. So if the leaf will be wet and the moisture will be more, then it definitely will cause the Manichuria in Achilles. So now we will come to the next question, question number 102. That is, Puccinia graminis pitici cannot cause infections on oat because. So as you know that Puccinia is a rust fungi and Puccinia graminis pitici doesn't cause infection on oat because it is a non-pathogenic. So it doesn't cause the infection on the oat. Puccinia graminis pitici only cause the disease on the wheat, black rust of wheat or black stem rust of the wheat. So now we will come to the question number 103. That is, conidia of the I.A.R.I graminis germinate in the low moisture because options are it has adapted to the stress condition, it has its own moisture, it contains lipid layer in the outer wall of the cell, or it absorbs the moisture from the atmosphere. The answer is it has its own moisture. So now we will come to the question number 104. That is, some bacterial strains produce proteinaceous antagonistic substance that are lethal to the other strains of the same or closely related species are called. Options are biotroph, bacteriocins, bacterioalleles, and bactericides. So the answer is bacteriocins. Now we will come to the question number 105. The process of identifying the plant disease is called. Options are detection, analysis, diagnosis, and inoculation. The diagnosis is the process of identifying the plant disease. So the option is C. Now we'll come to the question number 106. It is a very common question. Who established that the nucleic acid alone in the plant vessels is the infectious agent? Options are Stanley, Greer and Sermon, Borden and Peary, and Gibbs and Harrison. The answer is Greer and Sermon. Option number B. So now we will come to the next question, that is question number 107. An example of the mycorrhizal fungus is options are Penicillium, Pizzaia, Glomus, and Iricipi. The Penicillium, Pizzaia, and Iricipi are not mycorrhizal fungi. The answer is Glomus species. Now we come to the question number 108. Fungi belonging to which of the order are obligate parasites? What do you mean by obligate parasites? Obligate parasites are the parasites which derive their nutrition from the live host. So the agaricales, tephrinales, and australaginales, all these are not obligate parasites. The answer is plasmatophorials are the obligate parasites. Option number D. The question number 109. The example of autosis rust is the answer is linseed rust. The linseed rust is caused by the Melamsoralinae, and autosis rust example is linseed rust. Now we'll come to the question number 110. That the teleotospores are many celled in which of the genus? Options are Puccinia, Paragibedium, Euromyces, and Eurocystis. The answer is Paragibedium. Now we will come to the question number 111, that is blighted cast. A predictive model was developed for the epidemics of options are A. Early blight of potato, early blight of tomato, late blight of potato, and paddy blast. The answer is late blight of potato. Now we will come to the question 112, cross protection. The cross protection was given by the McKinney in 1929. The question is, the cross protection is effective in controlling which of the following virus in the citrus? The options are Badana virus, Tobago virus, and Mataran virus, and Clostridium virus. So the cross protection was given when the Tobago virus. Now we will come to the question number 113. If the resolution of human eye is 0.2 mm, then the magnification of the object will be 1x. If the optimum resolution of the light microscope is 0.2 micron, what will be the magnification? 1000x is the answer. So the Giemsa stain is used for staining. The options are Mycobacterium species, spores in bacteria, Rickettsia and some protozoa, and bacteria flagella. The answer is Rickettsia and some protozoa. Now we will come to the question number 115. Which of the following enzyme is not used in the ELISA techniques? The options are penicillinase, alkaline phospholase, protease, and horseradish peroxidase. The answer is protease enzyme. As a routine buffer used for grinding of the virus infected material is options are sodium citrate buffer, potassium acetate buffer, and potassium phosphate buffer, and sodium phosphate buffer. The answer is potassium phosphate buffer. So now we will come to the question number 170. Vemacia tabaki is commonly known as white fly. So the virus vector is also popularly known as options are sweet potato white fly, greenhouse white fly, potato white fly, and tomato white fly. The answer is sweet potato white fly. Question number 118. The aphid specificity of virus is located in options are N-terminal part of the port protein, C-terminal part of the port protein, nucleic acid binding protein, and movement protein of the virus. The answer is N-terminal part of the port protein. The common range of sedimentation coefficient for viruses are options are 100 to 200s to 500s, 480s, more than 1000s. The answer is 100 to 200s. Now we will come to the question number 120. The measurement of the color development of chromogenic substrate, that is p-nitrophenyl phosphate, for ELISA and spectrophotometer is done at, the answer is 405 nanometer. Now we will come to the 121. The rod-like structures of potato spindle tuber virus measures. The options are 20 nanometer, 50 nanometer, 25 nanometer, 100 nanometer. The answer is 50 nanometer, option number B. The three non-translated region of many viruses plays a role in options are movement of RNA, core protein assembly, initiation of the negative strand RNA synthesis, and folding of RNA. The answer is movement of RNA. So we were discussing the questions. Now we will discuss question number 123. Plant virus database of WIDE is located in which country? USA, Japan, United Kingdom, and Australia. The answer is Australia. The plant virus database WIDE is located in Australia. Now we will come to the question number 124. Screens with openings of which of the following will keep insect vectors out of the screen? Insect vectors out of the greenhouse, that is 30 to 60 mesh, 60 to 100 mesh, 100 to 200 mesh, and 200 to 300 mesh. Answer is 30 to 60 mesh. Gram positive bacteria belongs to the group and options are Brassicioles, Firmicutes, Tanigules, Meadowscutes. The answer is Firmicutes. Which of the following disease is caused by the fastidious bacterium? The options are citrus canker. Citrus canker is caused by the xanthomas. from pastures. Sativa citri. The blight of Paris caused by Xanthomonas oryzae. Cabbage rot and citrus green disease is caused by the fastidious bacterium. So option number D is the answer. Now we'll come to the question number 127. The diameter of the perforated filter with the laminar flow is about 0.2 micron. Laminar flow is a chamber that provides aseptic environment for the maintenance, working, and doing the laboratory work, culture of the pathogens, and free from the contamination. Now we'll come to the question number 128. The extra chromosomal DNA in bacteria may code for the host degrading enzyme, synthesis of antibiotic, host specificity, and disease induction. The answer is disease induction. Question number 129. Acid fast staining is used to identify options are Mycobacterium, Xanthomonas, Cyanobacterium, and Bacteriophage. The answer is Mycobacterium. Now we'll come to the question number 130. White flies transmit plant viruses in options are non-persistent in persistent manner, non-persistent in persistent manner, semi-persistent in persistent manner, and only persistent manner. The answer is semi-persistent in persistent manner. So question number 130. The objective type questions are completely solved. Now we will come to the matching type questions. Question number 131 to question number 140. All questions carry equal marks. Choose the correct answer A, B, C, D, or E for each sub questions 1, 2, 3, 4, 5, and enter the choice with a circle by shading with a pencil on the owner and associate as per the instructions given on the associate. Now we will solve the matching type questions. So the question number 131, the one is Radiocutaneous solenoid. So the Radiocutaneous solenoid matches with the Thanatophorus cucumeris. The Thanatophorus cucumeris is the perfect stage of the Radiocutaneous solenoid. Now we will come to the Fusarium molliforme. The perfect stage of the Fusarium molliforme is Gibberella fujifori. That is option C. And now we will come to the Bipolaris oryzae. The Bipolaris oryzae is perfect stage Pucciloborus neovenus. That is option D. And now the next is Phaseolus personata. The option of this answer is Mycoscarella burkii. Option number B. Now we will come to the question that is Trichorama viridis. The option C of the answer is Haplocaria rufa. That is option number A. So question number 131 is solved. Now we will come to the question number 132. Loranthus. Loranthus is mostly found in mango trees. The answer is mango tree. Question Striga. Striga are the root parasites. So the answer is anagamic root parasite. Option number C. Now we will come to Allomyces. Allomyces, the answer is alteration of generation. That is option B. Next, Olpidia. The angiogamous, isogamous, anisogamous lamogamous Olpidia. And the Pythium, options are metalaxial. The Pythium can be managed by the metalaxial spray. Now we come to the question number 133. These are the predicting models, forecasting models. EPDM. The EPDM was made for the alternaria solenoid, forecasting for the alternaria solenoid, always light. And now next is EPVEN. EPVEN is made for Venecheria iniquilis, scab of apple. Now the next is epidemic. The epidemic was made for the Puccinia gravis staphylococcus. The next is epicon. The epicon is Helminthsporium maedis. Option number E. And mycos for Mycoscarella. Option number A. So question number 133 has been solved. Now we will come to the question number 134. Amellose. The local lesion assay was discovered by the Holmes, and the cotton plug was given by the Schroeder HRP gene was given by the Stegwis, and AVR gene was given by the Lindergren, and MLO was coined by the Roy et al. Now we come to the question number 135. First generation fungicide is copper fungicide. Second generation fungicide is oxen. No, the second generation fungicide is oxanthin. Third generation fungicide is metalaxyl, and new generation fungicide is strobilurin, and fungicide comicicide is sulfur dust. Now we'll come to the question number 136. Copyrhinus species, it's a weed of the mushroom, and Septoria species is the defoliation of the tomato. The option number A. Alternaria alternata is the muriform conidia. The conidia are muriform. The Xanthomas campestris or Pledogoni is the vascular filter granarium, and Botratis cinerea is the grey mold of chickpea. Option number D. Now we will come to the question number 137. Cucumber mosaic virus. Cotton leaf curl virus, and potato leaf roll virus, and rice tumor basiform virus, and tobacco necrosis virus. So the cucumber mosaic virus, the cucumber mosaic virus is RNA 5. Cotton leaf curl virus is a nanovirus like molecule. Potato leaf roll virus is Potato belongs to the Luto virus. Rice tumor basiform virus belongs to the dependent transmission, and tobacco necrosis virus is satellite virus. Now we'll come to the question number 138. That is a rigid rod. So these are the rigid rod. The example of the rigid rod is cucumber green motor mosaic virus. The answer is of a rigid rod. The answer is cucumber green motor mosaic virus. Now we'll come to the question number bacilliform. The cocoa solar shoot virus has the bacilliform in shape. Now envelope shaped is groundnut necrosis, and the flexious rod is cucumber papaya ring spot virus. Papaya ring spot virus, flexious rod, and isohydron is cucumber mosaic virus. Now we'll come to the question number 139. The M.J. Narasimha have written the book Control of the Cholera Disease of Arcanaut. The answer is cholera. And B.L. Chona, the answer is written the book Red Rot of Sugar Cane. B.L. Chona. And now the B.V. Mundukar established the IPS, Indian Pathological Society. J.P. Verma has written the Bacterial Blight of Cotton, and P. Sadasivan are feasible of the cotton. Now we come to question number 140. K.C. Burden with M. Keeney has given the cross protection. E. Luria has given the bacteriophage. J.C. Walker has given phenolic resistance, and J.C. Craigie has given the sexuality virus fungi, and Borden has given the virus nucleoprotein. So the matching type of question has been solved. So this video will be very informative, and those who students are preparing for their PhD and are the first thing you must see, this question will be very helpful, and it is a format type.